Assalamu alaikum viewers, welcome to my channel Genome Study. If you are new on my channel, please subscribe and press the bell icon so you will get the notification of all the new videos. But don't forget to like this video. This is the second video about gene cloning, and in this video we will know that how we can select our gene of interest on which we want to do our work. There are different methods of gene selection on which you want to work, but it depends upon the project on which you are interested or the project that were running in your lab. You but first that you must select the attributes that on which attribute you want to work, either you want to work on the metaboloids or stress physiology or plant growth related functions. First you must have to clear that on which type of attribute you want to work. There are a lot of here is only I have given the example, and only these are the broad and that metabolic stress or plant growth. And after selecting this, then you have to choose that what type of genes you want to work. Either you have selected transporters or transcription factors or functional genes. Here I give the example about the metabolites. For example, I am selecting I want to work on the metabolites, and the gene that I have chosen is the enzyme, and from enzyme that is the transferase enzyme. But you can do according to your object and method for selecting all these genes is same for all that. What type of your objective is, what you are choosing for the genes, but the method, this method is same for all, you can apply it. And for in this case I will give you the example about the metabolites. By narrowing down, in metabolites there are different types, so I have selected the flavonoids. And then in transferase enzyme there are a lot of transferase enzymes, but further I have narrowed down it to, I have choose the Malonyl transferase enzyme, and which type of crop is soybean. So after selecting all these, so we can choose or find out or figure out the genes easily. First attributes, that is the metabolites. In metabolites we have selected flavonoids, and then in genes, that is the transferase enzyme. In transferase enzyme the Malonyl transferase, and the crop is our soybean. After selecting all these, you must know the detailed study about that gene that you have selected. For example, here I have selected the Malonyl transferase enzyme related gene, so I must know about it through the literature. I will study it. So in this video I will tell you about only about the method of selection of the gene for our project. How we can select it? Let's start. So open the NCBI, select here PubMed, and write here Malonyl Malonyl transferase, and then search it. So here it give the result that here is a phenolic glucoside malonyl transferase gene BTM81. This is the verticilloid NAT1 and malonyl transferase. And here you see that there are different type of malonyl transferases, but as we have talked about that we want to work on the flavonoids, so here more specify it, so we write it flavonoid malonyl transferase, then search it. Because in malonyl transferase there are different type of others also appears malonyl transferase, so we have, but we only need the flavonoid malonyl transferase, that we want to find out the gene that only work on the flavonoid, so flavonoid transferase. So we more specify through the keywords that flavonoid malonyl transferase. So here it comes that the molecular characterization of flavonoid malonyl transferase from either stiva it has come, and then melanated flavonoids, glucosides, two flavonoid specific malonyl transferases. These are all that flavonoid, but you try to find it that you can find in the closest species that we are working on the soybean. So you can find out in the like if soybean belongs to legume, so you can try it as a leguminous crop. Here we have also find that, oh, here is the soybean seeds, glucoside transferase and malonyl transferase from soybean. Someone also have identified a malonyl transferase from the soybean, but it is not necessary that only one the soybean genome have only one malonyl transferase. There are a lot of malonyl transferases that work on the flavonoid transferase. So we can also take it. It is also a very good sign that it has the functional transferase genes. So we can take this gene also, and then it is easy. We can find out the closest of this one, and then we can use it. So we can all open this, and also another one, this one, Gracin Max, and here flavonoid Medicago. Medicago is also belong to legume, so we can also take off this. So these are, I think, though two or three examples is enough. So we can find out here that he what name it gives to the malonyl transferase in the abstract or in the result. You can find out then here it shows that the malonyl transferase is here. Malonyl transferase is MT in Resin Max, and here it gives the name. This is IF GMIF7MAT, and here maybe this is a made. This is a transferase made transferase. So we can here search again their sequences, protein sequences. So back. So here we can select the protein, and MTR only Malonyl flavonoid Malonyl transferase in soybean. Search it. So here it gives the result that is MT2 malonyl transferase, no, or this is the malonyl transferase, this is malonyl, but we don't know that what was that one. So it is better that we have to find it with this name. This is the MT7, GMMT7. So here we can write it its full name, GMMT7. So here its name came that GMMT7, this one, and this is also IF7MAT, this one. So these both are came, so we can open its FASTA file and open the folder in which you want to work, and here I have already downloaded the sequence of that. This one is MT7, this is the IF7 MAT, and these were the Medicago MATs that I have found from the Medicago. You can find all these from this NCBA through this way. If you didn't find through this name, then it is better to go to this paper and find out their transcript ID. I have also told you in one of my genome wide study series that how we can select that genes for phylogenetic tree. You can also apply that method to find out that genes. So after that selecting these malonyl transferase genes, we need to blast them in the genome of soybean. So how we can blast it? For this we need the data of soybean. I have already downloaded it from the phytosome. You can also download it from the phytosome or any database available according to your crop. And how to download this, I have already made the video in one of my genome wide series. You can follow that and then also download that data. So for that, we for that blast we use the TB tool. So open the TB tool. So open the TB tool, go to blast and to sequence file, and here subject sequence we this one, the protein sequence file. We put this protein sequence file in the query protein. We have this file, the MAT files, and we can also take the result here. So right here, empty list, melon transfer list txt. From here you can select the table, and from here number of hits, we only need the 2 to 3 best matching genes with our that query protein. So we select only 5 and then click start. So here finish, jump to the no. So here is the list of genes that best, that gene that were found on the basis of our query proteins. So this is the Medicago gene, MT, M84, and these are the best 4 related genes. And here are the M85, and these are the 4 related genes. And then this is the MT7, and this is the 100%. It means this gene is already someone have worked it, so we didn't take this one. And this one, you see this gene has about 87% identity with this gene, so it is most probably that it also has the function of this gene, so we can take also this one. And this is IF7, and this is also, it has the 100% identity with IF7 MAT, so we also didn't take this, but it has 13G056100, and oh means MT7 and IF7 MAT was the same gene. The two groups have worked on the same gene, but we take their closest one. So we have to take these genes, select all, copy, open that Excel file, and paste here, and then we delete this. So this one we have. So you see that this gene have 100% identity with that one, so it means that this MT7 gene is this one, so we have to delete this one, we discard it because we didn't take this one. And IF7 also. So these are our best genes that were give the best hit to this already characterized genes. So here if we have the means duplicate gene, then we can also delete them. Control X, and here data, and remove duplicate. So we get this about 15 unique genes, 14 unique genes. So now further we have to do their selection on the basis of their expression level. So here is a file that contains the expression of all the genome data. And how we can find out these files from the expression data, that RNA, if you have the RNA-seq file about FPKM values or expression data, you can use that one, or you can also download it from the GEO, NCBI GEO. How to download this? So you can find out the link at the end of the video or in the video description. You can follow it and also download that expression data. So we can use this file to find out the expression of these genes. So here go to the others, table row manipulation and input table. So this I put that data and use this. So here you see that there is no dot p or dot one or nothing else. So we can select this, selection done, and where we want to save our that expression data. So here empty expression.txt, and here set the that IDs. So that ID that we have selected. So here you see that it has dot one dot p dot one dot p. So we need to remove this dot one dot p. So control H. Here you write dot one dot p, replace with nothing, and then replace all. So our IDs becomes according to our file that we have used in the TB tool, and then you can paste the your IDs here and then start. Yes, that column, and no. So our results have come. Empty expression. So here are the expression of these genes. Select all, copy, and then we need to paste here. So expression, we have other expression of all that gene. So we have to remove the other columns that only we need that ID. So we don't need these columns. So we have to remove, and this one. This is the expression in pod and root here like this, but this is the FPKM. This is the confidence low and confidence high, confidence low, confidence high, and pod status. So we didn't need this one, so we have to remove this. We only keep the FPKM value, so we have to remove these columns, these all columns. So we have removed all other columns, only except keep the column that have the FPKM values, and these are the different parts or tissues of the soybean, that is pod, root, leaves, root hair, nodule, seed, stem, flower. And as we know that the in soybean the flavonoids were more have in roots, seeds, and flowers. Flavonoids in soybean have the roots, root, seed, and flower. So we can check that which gene have the highest expression in root, seed, flower, and also the important factor that was the nodules in legumes. So these four tissues we will take for our observation of the expression data, that which gene have the most expression in that tissues. But you can check your gene according to your gene's function. If you are working on the stress, then you can select the expression that on the response of stress, how which gene expression is higher. You can select on the basis of that, or that other, if you are working on the another any other attributes, so you can select according to that attribute and in which part of that attribute is important. So you can select or check the expression of your genes, selected genes, or find out genes in that parts, and then you can select it. But for me, the important parts for flavonoids is the root, nodule, seeds, and flower. So I will check the expression of my gene in these four tissues. So the gene that has the highest expression in that tissue, so I will take that one. So here you can easily is better is that you can select this and then go to the conditional formatting, and here you can select the colors. So you can see here that this gene, you can see that this gene have in root here have the high expression in root here, and this is a seed, and flower is hello, but these tissues have high expression. So one of my gene that I want to select this one. So for that functional characterize, you must select at least three genes, 3 to 4 genes for the characterization, and on that 3 to 4 genes, that one or two must work, and it didn't waste your time. And another one I must select is you can see in flower if any have this gene. This gene have in flower specific, right? It has a high expression in flower, and if any have the nodules, here is the nodule. This is already selected, and one of that gene you see that this gene, it has a higher expression in the only in the that SAM as compared to the other gene, other tissues. So you can also select this one, and this gene like this one have also have the high expression in flower, and somehow have the in nodules and root, but it has also in the roots higher expression. So you can also take these, so you can take these 4 genes for your study. So on the basis of expression data means from that I want to select these in 4 genes that's for my project. But if you are working in any other aspect other than the metaboloids, the such as the stress related genes, then you can apply the same method to find out the genes and download the stress expression data from GEO, or if you have the expression data or RNA-seq data of you in your lab, so you can apply that, and then you can check that the expression of that gene, and then you can select that 2 or 3 genes and carry out your work. So in this way you can select your gene for your project. So further we will know that how we can download the or get the sequence of that gene, selected genes, and how we can prepare the primers, and how we can do the experiment on these. All this we included in this series. Stay tuned. It's all about this one. If you have any query, please comment. Don't forget to like. Thank you.